When we discuss blood vessel physiology, we're discussing blood pressure, of course, is the big topic, but also the velocity of blood flow or the speed of blood flow. And also, a separate topic would be circulatory shock. We're going to divide up our paper right away into the three main topics. At the top, basically, the entire top half of the paper is going to be discussing blood pressure. First of all, we're going to describe what blood pressure is, and then we're going to talk about regulating our blood pressure. We're going to have a separate topic for discussing the velocity of blood flow or the speed of blood flow through separate tissues of the body. And then lastly, we're going to be discussing circulatory shock. Well, I should actually say that's the first thing we're going to be discussing because it is kind of a special and unusual topic that's sort of separate from the other ones. Now, circulatory shock. We're going to start off with describing the first example of circulatory shock, which is called hypovolemic shock. Hypo means low and volemic means volume. So hypovolemic shock means that your circulation is in low volume, and this is usually due to an extreme fluid loss of some kind. Now, I bring this up because the actual definition of circulatory shock in general, like the umbrella topic, is that the blood vessels are inadequately filled. Usually people think about hypovolemic shock as circulatory shock. They think, oh, okay, you've lost a lot of fluids, you've lost a lot of blood, something like that. That's circulatory shock. But I want to impress upon you that there's actually three different ways that this can occur. Hypovolemic shock is only one of them. Another way that it can occur is through vascular shock, which would be extreme vasodilation. And then the third way, which is probably the easiest because we've talked about it before, is cardiogenic shock, which is simply a pump failure. So a pump failure means that your heart is not functioning properly and is not pumping enough blood out. We've discussed this before. Congestive heart failure or cardiac arrest would be the examples of pump failure or your cardiogenic shock. So let's go back to our first idea of hypovolemic shock. The first way, then probably the most common way that we discuss hypovolemic shock would be through a simple hemorrhage, a heavy blood loss. You've damaged some kind of blood vessels in your body and you're either bleeding externally or perhaps you're bleeding into your body cavity. A common way that this would occur internally would be through a severing of the splenic artery, which happens to be in a vulnerable place without a lot of protection from bones and also happens to carry a heavy flow of blood. And so when a person is in a car accident and, for example, the steering wheel juts into their abdomen, oftentimes the physicians will be looking for bruising in the region of your spleen, which would be your your left upper quadrant of your abdominal pelvic cavity. Other ways that you could lose a lot of fluid from your bodies would be simply through heavy diarrhea or vomiting. And also, this is one of the things that we are concerned about with burns. We talk about looking at how much of a body's surface is burned because one of the things we're quite concerned about is fluid loss with very widespread burn occurring. All right, let's take a look at our vascular shock. There's a number of ways that we can experience a vascular shock. A vascular shock, the basic idea here is that the blood vessels are popped open. They are wide. This vasodilation means that there just isn't enough fluid. Like your fluid levels have remained stable at about 5 liters, which is typical. But the dilation or the opening up of those tubes means that there isn't enough blood being passed around with enough pressure, I guess, or enough filling. The tubes are not filled, and therefore the blood is simply not going to be efficiently passed around the body and is not going to be efficiently transported. And so a common way that we think of this occurring would be through anaphylactic shock. Usually we think about anaphylactic shock as being an allergic reaction associated with the closure of the bronchi or the airways going down into the lungs, but it turns out that is also paired with vasodilation. Now, the next type of vascular shock would be a neurogenic shock, so that the areas of the brain that are involved with regulating that blood vessel diameter, such as the sympathetic nervous system, might be damaged. The next type is called the vasovagal syncope, which is a standard, basically what we oftentimes call shock. A person is in shock when they've experienced an extremely stressful event, and oftentimes they will faint as a result of this. The blood drains from their face. They become white in the face. and oftentimes they will faint. And then the nice thing about that is that the blood vessels will usually kind of right themselves because when a person is passed out, they're not as stressed out by the incident. Now, the next type of vascular shock is quite serious, septicemia, or your blood going septic. It's basically blood poisoning, and we'll be talking about this more when we talk about the lymphatic system, actually, in the next lecture. And it's quite serious. And then lastly, we have transient vascular shock, which is a situation where a person just briefly experiences an extreme plummet in their blood pressure, an extreme vasodilation, but it's usually just a very specific situation. For example, a woman who is pregnant and has just lots and lots of fluids in her body, if she's lying down for a long period of time, she should be careful getting up out of bed because once she stands up, she might experience that extreme blood pressure drop, and she might feel dizzy and might actually collapse as a result or faint. The same thing will happen typically when a person is in a hot tub for a long period of time. The hot tub is going to cause a dilation of the blood vessels. And when the person stands up out of the hot tub, especially if they're, for example, outside in the cold or something like that, all of a sudden the blood vessels are going to try to snap together. And as a result, again, you could have that drop of basically the blood maybe not going up to the brain properly. The brain is supposed to get a certain amount of blood every minute, and that blood is not being shunted properly to the brain, and therefore the person might still feel temporarily dizzy, or they might actually faint or collapse. Right, so let's move on to blood pressure. We're going to draw an XY axis here. On the y-axis, we're going to be drawing pressure in millimeters of mercury. It's a typical way that we measure pressure in multiple aspects in this class. But I wanted to take a moment and describe what blood pressure actually is, because I think it can be a little bit misunderstood. So we just want to make sure we're all on the same page with this one. When we look at blood pressure, it's helpful to think about any blood vessel in your body. It's usually going to be an artery, but just any blood vessel technically can experience a little bit of blood pressure. And what the blood pressure is the pushing of the blood, hydrostatic pressure, against the walls of the blood vessels. Basically, a pressure from the inside out. And so as the blood makes its way through the blood vessel and it's following the diastole or the resting phase of the heart, there's going to be a little bit of push against the walls. But as these guys experience a surging of blood coming from originally from the systole, the systolic phase of the cardiac cycle in the heart, and the ventricles have pushed and pumped a big surge of blood out, and then this is going to put more pressure against those walls. So depending on where you are in the cardiac cycle or where these vessels are following it, these could be experiencing just a little bit of pressure pushing out against the walls or a great deal of pressure. And that's why we actually have two numbers when it comes to blood pressure. It just depends on where you are in the cycle. So we have our diastolic number and our systolic number. And especially when you're very close to the heart and you're in the artery side of things, the arteries, the aortas, you're going to be measuring two quite different numbers because the systolic number and the diastolic number are separate. The systolic number is typically about 120 millimeters of mercury, and the diastolic number is typically about 80 millimeters of mercury. But it does, as you can see, the further you get away from the heart, there's a kind of a precipitous drop of blood pressure from these high numbers almost down to zero. So right in the middle, we're going to label this first because it's probably the most interesting area is the capillaries. The capillaries are going to be where you're going to have the biggest drop in blood pressure. But of course, you're going to have that separation when you're more toward the aorta and artery side of things. And then the further you get away from the heart, or the further you get away from the ventricles, I suppose, like for example, the vena cava or the veins, you're going to have pretty low blood pressure. So you're going to be approaching zero. So hardly any pressure in those blood vessels at all. Remember that in order to have pressure in the blood vessels, you have to have some kind of a muscular contraction. And remember that the veins really don't have muscles. They do not have much of a tunica media, and therefore their blood pressure is quite low. They are meant to just receive blood and not put any pressure on it. Right, so there's a number of different ways we can discuss regulating blood pressure, but I figure the easiest way to start off with is just to put everything into a framework. when we want to either lower blood pressure, which we're going to be drawing in green, or raise blood pressure, there's really only two factors we have to pay attention to. If you understand these two factors and kind of put everything into that framework, a lot of the details will become a little bit easier. So there's only two things that we can actually control. We can control blood vessel diameter, either through vasoconstriction or vasodilation, and we can control the amount of blood volume that we have. So let's talk about that blood vessel diameter for a moment. I always use the hose analogy. The idea being that if you have a garden hose and you want to make the water squirt further and put more pressure on it, we all kind of intrinsically understand what we do is we put our thumb over the mouth of the hose, and changing the diameter of the mouth of the hose will cause more pressure to be exerted on the water, and the water will squirt. So that's exactly the same idea here. When you vasoconstrict a blood vessel, you are closing off some of the diameter, and this makes the water be exerted under a great deal of pressure. And this, the opposite of true is true, of course, of vasodilation. If you want to open up your blood vessels, that's going to decrease your blood pressure. On the other hand, I think this one's a little bit easier to understand. Blood volume, if you have the same tubes and you put more fluid into them, then you will have a greater pressure against the walls of the tubes. So more blood volume, higher blood pressure; less blood volume, lower blood pressure. And I think the one part of this that we have to kind of wrap our minds around a little bit is remember that what gives the blood its volume is water. So if we can control the amount of water in the blood, this in turn will either raise the blood volume or lower the blood volume. So watch out for these two aspects: vasoconstriction and vasodilation, and anything that would either raise or lower your blood pressure, your blood volume will in turn start to affect the blood pressure as we are stipulating here. So yes, there's a lot of details coming up. There's quite a lot to take in, especially when we talk about the hormones involved here. There's a lot to think about, but if you put everything into this framework, I think it's all going to fit together much more readily. So let's begin with what we call fast changes to your blood pressure. These would be involving the brain, and specifically the brain stem. These would be short-term changes, kind of temporary situations, and they would be very fast. Now, the center for blood pressure change is going to be in the brainstem and the medulla oblongata particularly. We're also going to go ahead and label the spinal cord here because that's going to come up in a moment. The neural changes are involved pretty much exclusively in either vasodilating or vasoconstricting the blood vessels. Remember that these will have a say in how the sympathetic nervous system will work. And so remember that the sympathetic nervous system, these neurons coming from the spinal cord, will in turn cause it, they will send adrenergic innervation into the tunica media of the blood vessels, and in turn cause vasoconstriction. And so that's how it works. You can send the impulses to vasoconstrict, or you can stop sending the impulses to vasodilate. And so we have this nervous impulse here, and it all centers into the medulla oblongata. So as long as you keep that in mind, then we're able to think about there's three different main inputs that come into the medulla, and therefore the medulla can make decisions about what to do about how to change the blood pressure in keeping with what is needed in terms of what kinds of information is coming in. So we start off with the hypothalamus. The hypothalamus is the part of the brain that is sort of responsible for lots of body things going on, body temperature regulation, your hormone regulation, and things like that. And so the hypothalamus is going to have an idea of what's going on inside of the body, and will therefore be able to put some input into the medulla in that way. Secondly, we have baroreceptors. The root baro means pressure. So this is simply measuring the actual pressure, as we said earlier, that is being exerted against the walls, especially of the arteries that are close to the heart. So baroreceptors are little sensors that sense pressure changes to the artery walls close to the heart. And then the third type of input here are going to be chemoreceptors. Chemoreceptors are going to be found either in the measuring the amount of chemicals in the blood, or, and again, these are going to be arteries close to the heart, or also in the CSF, which is your cerebral spinal fluid. And so there, we're measuring things like hydrogen ions or carbon dioxide levels or simply the blood pH. All of these three things are related to each other. And what is happening inside of the blood is also closely reflected in the CSF. And therefore, either of these fluids will do to be measured and then send that impulse again into that medulla where adjustments can be made. Now let's move on to our hormonal regulation. This kind of this family of hormones that are responsible for various aspects of what's happening. Remember the framework. Remember vasoconstriction and vasodilation and blood volume. So the first of the hormones we're going to be dealing with, we're going to draw in green, and this is ANP, atrial natriuretic peptide, released by the heart, by the atrial walls. And the ANP is going to do a few things that help to lower your blood pressure. One of them is it's kind of a weak vasodilator, but significantly it's able to increase the amount of sodium ions that are released into the urine. So the target here is going to be the kidneys. And I just wanted to put this out there because we discussed this dictum when we discussed the endocrine system, but I want to make sure we're really clear on this. Where sodium goes, water follows. Where sodium goes, water follows. So if you release sodium into the urine, you're also going to be releasing water into the urine. And when water enters the urine, that means that it has left the bloodstream, and therefore you have lowered your blood volume. So this ANP, atrial natriuretic peptide, is a hormone that actually lowers your blood pressure. This is the only one that I know of. There's not that many hormones that actually lower your blood pressure. Actually, there's several hormones that raise your blood pressure. So let's go to that side of the story now. We're going to be discussing the angiotensin-renin story. And we're going to start off with an inactive structure called angiotensinogen, which is released by the liver. Angiotensinogen, under certain conditions, will convert into another substance called angiotensin 1, which will be further converted into angiotensin 2. Now, angiotensin 2 is an extremely powerful raiser of your blood pressure through various means, which we will discuss in a moment. But first of all, let's discuss how these things actually can get converted from one to the other. Angiotensinogen is converted into angiotensin 1 using a hormone called renin, which is released directly by the kidneys. Angiotensin 1 then is further converted into angiotensin 2 using ACE, which is angiotensin converting enzyme, which is released by the cells of the lungs. Now, angiotensinogen is something that is, the inogen there indicates, the root inogen, I-N-O-G-E-N, indicates that it is an inactive structure, and therefore you actually need renin to be released to have any kind of activity at all. Once you get to the level of angiotensin 1, angiotensin 1 does have some level of ability to raise your blood pressure, but it's only when you get to angiotensin 2 that you have a really powerful way of raising your blood pressure. So angiotensin 2 is a big part of the idea that people have high blood pressure, generally speaking. Angiotensin 2 is a big part of that story. So if people have a lot of angiotensin 2, they might actually have really high blood pressure called hypertension, and in that case, you need to think about what you're going to do about it. Well, let's talk about why angiotensin 2 is so powerful. There's actually 4 different things that it does. First of all, it will enhance your thirst mechanism, which will encourage you to drink water, which will in turn increase your blood volume and therefore increase your blood pressure. Angiotensin 2 also is a powerful vasoconstrictor, therefore using these two means, by raising your blood volume and constricting your blood vessels, it increases your blood pressure on its own. But it also enhances the production of 2 different hormones. For example, it enhances the production of ADH, or antidiuretic hormone, from your posterior pituitary lobe. And antidiuretic hormone, ADH, is a weak vasoconstrictor, but its main method of working is by increasing the reabsorption of water directly at the kidney tubules. We'll be discussing this more down the line. Kind of a cool little story there. So when you reabsorb water at your kidneys, that means you're not releasing the water through urine. You're retaining it in your body, therefore raising the blood volume of water and increasing blood pressure that way. And lastly, angiotensin II will increase the production of aldosterone, which comes directly from the adrenal cortex. Aldosterone is one of those mineralocorticoids, those steroid hormones that comes from the adrenal cortex. Aldosterone pretty much only has one role, but it's a powerful one. It will enhance the reabsorption of sodium ions at the kidneys. Remember, where sodium goes, water follows. So when we reabsorb sodium, we also reabsorb water, and this will help us to retain more water, which increases our blood volume, which increases our blood pressure. It's worth taking a moment to admire the work of the angiotensin II, if you will. If you want to raise your blood pressure, you are sure going to want angiotensin II to be part of that story, because it has, as you can see, 4 different powerful methods of increasing your blood pressure. Oftentimes, though, as humans in this day and age with the particular diets and lifestyles that we have, oftentimes people will suffer from hypertension or high blood pressure, especially chronic high blood pressure or long-term high blood pressure. And this is a bit of an issue because, for example, we talked about how it affects the heart, how it can damage the heart over time. The heart walls, they have to pump so hard against that high blood pressure in the arteries. The artery walls themselves will start to break down under high blood pressure conditions. And there's many organs of the body that will break down under these conditions, but a big one would actually be the kidneys. So we want to make sure that we are taking care of our body as best we can. So now we have classes of drugs that actually help to lower our blood pressure, because it is damaging to have hypertension for a long period of time. So one of the classes of drugs will be called ACE inhibitors. So an ACE inhibitor basically interrupts the pathway here. Instead of going from angiotensin 1 to angiotensin 2, we can partially block that conversion by blocking the presence of ACE, that angiotensin converting enzyme that brings angiotensin 1 to angiotensin 2 will be interrupted in the presence of these drugs, of this class of drugs, and therefore it will help to lower your blood pressure, especially if you are already suffering from high blood pressure. All right, well, our last category is blood velocity, and we're actually going to continue the picture that we had up above where we separated out the aorta, arteries, capillaries, veins, and vena cava, and we're going to continue those by looking at the picture down below. And we're going to go ahead and discuss the cross-sectional area in centimeters squared of each of these: aorta, capillaries, and vena cava. And specifically, since we're measuring this in centimeters squared, we can just put the numbers down there. And aorta, about the lumen cross-sectional area is going to be about 2.5 centimeters squared. A vena cava is about 8 centimeters squared. But the capillaries are 4,500 centimeters squared, which you're thinking, well, that doesn't really make a lot of sense because capillaries are tiny, and that is true. But what we are able to do is add all of them together, and if you add all of the lumens of all of the capillaries together, they actually have much wider cross-sectional area than either the aorta or the vena cava. Now, it turns out that there is an inversely proportional relationship between the cross-sectional area and how quickly blood will flow through that area. So the lower the cross-sectional area, the faster blood will pass through the area. So the aorta, blood passes relatively quickly at 40 to 50 centimeters per second, and the vena cava are kind of second at about 10 to 30 centimeters per second. But those capillaries, this is the most interesting part of the story. The capillaries, which have such a high cross-sectional area, are going to have the slowest passage of blood through there at 0.03 centimeters per second. Incredibly slow, and especially in comparison with the aorta or the vena cava. So the 2 follow-up questions to this are, why? Why do the capillaries have such a slow passage of blood? What is this inversely proportional relationship that we're discussing here? So this is where I discuss the river versus the lake situation. My students have I've always liked this analogy, and my students report that they like it too. So here it is. When you have a river, the river which is going through a small passageway, kind of a small tube, if you will, is going to rush along. But as soon as that river opens up into a lake and has a much wider area to go through, it slows down. And you know this to be true if you've ever been On a boat that's going through a river and then it opens up into a lake, it goes much slower once you hit the lake. And then once you get to the other side of that lake and you're going again into a river, say between a big gorge or something like that, and you will speed up again as you enter into that river. So again, the smaller the tube, the faster the velocity, the faster the speed going through it. Same with the blood going through a smaller versus a larger cross-sectional area. And the last question to be answered, which doesn't sound like a different question, but it is a different question. Why is it useful? Why is it useful that blood slows down as it makes its way through a tiny little capillary? Well, it turns out that this is the place, as you may recall, this is kind of the whole point of the circulatory system in some ways. You want to have the exchange. You want to be able to exchange things from the blood into the tissues. You want oxygen to get into the tissues. You want carbon dioxide to be able to leave the tissues and enter into the blood, nutrients to get into the tissues, the acidic metabolic waste products get into the blood. So you want this exchange to happen as efficiently as possible. So when we slow down the blood supply in these areas and the capillaries, which are the only exchange vessels, then we have the ability to exchange these things as efficiently as possible. This is the first time that we're coming across a filtration mechanism where we want to slow things down for as much exchange to happen as possible, but we are going to be coming into this same idea several more times. Whenever you want to filter something, whenever you want to exchange something, whenever you want to have good solid diffusion across a membrane, for example, then you want things to go as slowly as possible through those regions. And our body always sorts out mechanisms to allow that to happen as efficiently as possible.